Gives me absolute great pleasure and delight to introduce you to Professor Olaf Stuve. You can see he is from the neurology section and neurotherapeutics from the University of Texas in Dallas. We've known each other for over 30 years, but this is the first time since then that he's come to South Africa. So it's wonderful to have him here. We also have in the room his wife. She's a plastic surgeon and she's a wife, but also has her PhD in neurobiology. So there are lots of exceptional brains in the room today, and that very much excludes me. But it's lovely to welcome him to speak on multiple sclerosis. When I met Olaf many years ago when he was a medical student, he was already talking and thinking about neurology. And he was one of those people that you meet when they're young and you know that they're going to go places. So it's just wonderful to see what's happened with his career. I'm not going to go through the CV, but I think some of the things that came out as I read it that really is important to us is multiple awards for teaching, a lot of mentorship and training, and that's something we've been very interested in. The other thing is, of course, his bench to bedside approach to his diagnosis. So he's the true clinician scientist. And from the SAMRC perspective, we really value clinician scientists and have tried to grow that not only in the MRC, but also in the country and on the continent. And then the final and most important thing is that all of this is due to his internship, which was at Groote Hospital. So when he came as a medical student, he enjoyed Cape Town so much that he came back to do his internship with us for a year. So I think just with that, Olaf, a very warm welcome to you as well. We look forward to hearing all not only about multiple sclerosis, but we'll use a little bit of time at the end to ask you questions about your journey or anything else you'd like to add. So over to you.
Thank you, Liesl, for the very nice introduction, and it's a real pleasure and honor to be here, of course. And as you mentioned, I did my internship here 31 years ago. When you look, if you look closely, you can see a lot of happiness in that face. So these are my disclosures, but I really have no financial conflicts of interest with what I'm about to present to you. And these are my objectives. So essentially, I'm going to talk a little bit about epidemiology and changes in how we view MS phenotypes and new developments that allow us to diagnose MS very early and how that translates into new pharmacological interventions. And I have a lot of slides and I'm going to talk quickly. There have just been so many new developments really in the last 5 and really in the last 2 years that I think I shouldn't leave anything out completely. But again, I think I'm going to make it on time. So MS is the most common inflammatory disease of the spinal cord and the brain. As you can see here, more than 2 million individuals are diagnosed with MS. And in the US alone, there's 750,000 patients. Mostly, and that's true for many autoimmune diseases, young women are diagnosed with MS. There's a preponderance of 1 to 3 to 1 to 4 currently, and it's the major non-traumatic cause of neurological disabilities in young people. So it's got a tremendous socioeconomic impact for patients and also their families. This is a map of the world that shows you the prevalence of multiple sclerosis around the globe. And you can see immediately that there are substantial differences between, let's say, North America and Western and Northern Europe and other parts of the world, including Sub-Saharan Africa. Now, that's changing currently. And we know, of course, now genetically, there's no difference between people everywhere in the world. There may be some differences in environmental factors that explain what you're looking at here, but I think mostly it's really a problem of ascertainment. You know, it really takes very expensive methods to diagnose MS, including MRI scans, and some of these methods really haven't been widely available in parts of the world. This is work done by colleagues at Stellenbosch University, and you can see here that since the 1990s, both the prevalence and the incidence in Sub-Saharan Africa is actually increasing. And again, I think that's not because there's a true change in incidence and prevalence, but really a change in the ability to diagnose multiple sclerosis. And this is how multiple sclerosis looks like clinically, the disease course. So more than 90% of patients with multiple sclerosis are initially diagnosed with a relapsing remitting disease course. So they have a first clinical attack that's followed by remission, either complete or incomplete remission, and then in variable intervals they have more relapses. And eventually they no longer fully recover and they start to accumulate neurological disability. And one thing that's interesting is that eventually the number of relapses over time really diminishes and eventually completely goes away. But this accumulation of neurological disability actually accelerates. There's something called PIRA that some of you may have heard about. That's why I mentioned it, or progression independent of relapse activity. So going back to this slide, it was always thought that all the accumulation of disability is the consequence of having relapses, but that's being challenged right now. People have gone back to all the phase 2, phase 3 clinical trials and shown that even if you look 90 days after a relapse, there is occurrence of new disability. And some people think it's independent of a relapse, and I think that's somewhat debatable, and I'm not going to talk much about it, but I'm happy to discuss it later. I think most neurologists and neuroimmunologists think of multiple sclerosis really as a two-stage disease. And what that means is, again, early on, you have young people with relapsing remitting disease. And really, many of these patients now with pharmacotherapies live a fairly normal life. But then as the disease goes on, after 10 years, 50% of these patients with relapsing remitting MS will transition to having secondary progressive MS, where they, again, accumulate neurological disability. Many of my colleagues also refer to the early phase of MS as inflammatory MS, and the second phase of the disease as neurodegenerative MS. And I will talk about this a little bit. We do not know why patients transition from early inflammatory relapsing remitting MS to later neurodegenerative secondary progressive MS. We don't even know when this transition occurs. It's always a retrospective diagnosis, and it usually takes 5 years of somebody having transitioned before one is really sure that they now have secondary progressive MS. So we don't have any biomarker at this point that allows us to really determine when this transition occurs. I mentioned earlier it's an inflammatory disease, and really inflammation is visible in all stages of multiple sclerosis. So this, inflammatory MS versus neurodegenerative MS is somewhat of a misnomer because, again, you have inflammation of all stages of the disease. And what you're looking at here is an H&E stain. It's a picture of a post-capillary venule, and you can see that around the blood vessel you have this perivascular space that's loaded with leukocytes. So most of these cells are T cells, B cells, and myeloid cells, so dendritic cells, macrophages, and they're not supposed to be there. So if you would look at a healthy age-matched individual, you wouldn't have inflammation in the brain. It's really supposed to be sort of like an privileged organ where you really don't have these kind of immune cells. I should also say that we think all of these cells come from the bone marrow, so they're not CNS intrinsic. They migrated there to trigger or perpetuate inflammation. The other thing that we see in multiple sclerosis, of course, is neurodegeneration. And this figure here is the most cited figure in neurology. In history, it's by colleagues at the Cleveland Clinic led by Bruce Trapp. And you're looking at confocal microscopy, where a protein called neurofilament, non-phosphorylated neurofilament, is green, and the myelin sheath surrounding the axons is red. And you can see these round structures here on the panel of the upper left, and those are called onion bulb formations. So if you have an axon in the brain and it dies, it transects and the distal ends roll up, and that's what's called onion bulb formations. And you can see that a lot of these occur in areas of demyelination. And this is something that's abundantly present in multiple sclerosis. So this is an active acute MS lesions. And in 1 cubic millimeter, you have on average 11,000 transected axons. So it's really massive neurodegeneration, and that's something that we only recently have appreciated. Now, as I mentioned earlier, MS is a very dynamic area of research, and that is also true for pharmacological developments. So since I was an intern here at Grote where there were no approved therapies available for patients with multiple sclerosis, and in fact, we saw a lot of patients with MS in wheelchairs, young people. There are now 24 approved agents for patients with early relapsing remitting multiple sclerosis. And I just want to re-emphasize what I just said. All of these drugs are approved for early multiple sclerosis, because what they do is, and why they get approved, is they reduce the frequency of relapses. So that's important to keep in mind. So these therapies are available only for a subset of patients at this moment. And the dogma from the very beginning has been you treat as early as possible to prevent the accumulation of disability. And there has really not been any evidence to support this dogma until recently, but it's really biologically plausible. So that's really what people have been doing, at least in North America and in Western Europe, where those therapies are really widely available. So, of course, if you follow this dogma, then you want to diagnose multiple sclerosis as early as possible. You don't want to waste years before you start treating patients. So when I trained, you had to wait for 2 MS attacks to occur to make a diagnosis. You may have heard this term, dissemination in space and time, to make a diagnosis of MS. That means you have to have at least 2 clinical events separated by 3 months in 2 separate areas of the central nervous system. So you had to wait for that to occur. And in most patients, that took 2 years at least, that you wasted before you could really initiate therapy. 25 years ago, people said, we don't want to wait for the second attack. We want to make a diagnosis when patients have their first clinical event, and that's called a clinically isolated syndrome. And the reason we were able to diagnose MS already at that point was that MRI was introduced really in the 1980s and allowed us to demonstrate dissemination in time and space in somebody who only had 1 clinical event. And that is because you have these signal changes on the MRI. We can see this here on the left panel. They're periventricular, they're ovoid shaped, they're perpendicular to the lateral ventricles like Dawson fingers, which you may know the term. Dawson was a Scottish pathologist who first described them 150 years ago in autopsy material. So you can really see these lesions. And if they are not contrast enhancing, that means they're at least 3 months old. So if you get a scan with contrast and you have some lesions that are contrast enhancing in one area and some that are not, you have dissemination in space and time. So that was a real step forward. But in the last 10 years, people have started to think about diagnosing MS even earlier, so even before they have their first clinical event. And that's called radiologically isolated syndrome. And a colleague of mine at the University of Texas, Darren Okuda, was really, I think, the one person who has spearheaded this recognition. So essentially, somebody comes into your office, if you're a neurologist or a general practitioner or a pediatrician, and they got an MRI scan for something that's completely unrelated to an MS attack. So they have fatigue or dizziness, or maybe they hit their head. You get an MRI scan and it looks exactly like an MRI scan of somebody with multiple sclerosis. That's a radiologically isolated syndrome. And of course, not everybody can be diagnosed with that who has any sort of signal abnormality on the MRI scan. There are specific diagnostic criteria. But they are agreed upon, I think, universally among neuroimmunologists. So that's a step forward into diagnosing early. And we now have enough data to have some some idea about the natural history of radiologically isolated syndrome. What's interesting is that if you follow those individuals with a radiologically isolated syndrome, half of them within 10 years will develop multiple sclerosis. So it is a meaningful observation. There are also two clinical trials, phase 3 clinical trials that were placebo-controlled that showed that if you treat individuals with a radiologically isolated syndrome with drugs that are currently approved for multiple sclerosis, namely dimethylfumarate and teriflunomide, you prevent the onset of multiple sclerosis, or at least you delay it. I should point out that for radiologically isolated syndrome, there's currently no approved medication. The FDA is still not approving medications for individuals with radiologically isolated syndrome. And actually, it's somewhat of a controversy, at least in the United States, whether to treat or not. Because if you go back to this slide, 10 years after making a diagnosis of radiologically isolated syndrome, 50% of patients will develop multiple sclerosis, but 50% of them will not develop multiple sclerosis. So whom do you treat, right? Are you going to treat everybody and prevent MS in the ones that are at risk? But then you're going to treat 50% of people who will never develop possibly multiple sclerosis, and you're going to expose them to the side effect of the drug. So there's knowledge missing to really identify the ones that are truly at risk for developing multiple sclerosis. It becomes even more complicated because in the last 5 years or so, other colleagues have identified a prodrome of multiple sclerosis that occurs at the time patients are diagnosed with radiologically isolated syndrome or many years before that. And I actually had never heard of the term prodrome until recently. So just to explain it, it's signs or symptoms that occur before a diagnosis is established in individuals eventually diagnosed with the disease, in this case multiple sclerosis, and they're really very non-specific, as you can see on the slide here. So pain, headaches, fatigue, bowel, skin, bladder problems, so something that almost everybody experiences at some point in their life, so very non-specific. And in fact, in multiple sclerosis, it was thought to be there is no prodrome until recently, whereas in other autoimmune diseases like lupus and rheumatoid arthritis, prodrome is actually well established. Now, colleagues from Canada, where they have universal healthcare and medical data are collected, basically identified in their healthcare record 14,000 people with MS and did a matched age-matched control study with 72,000 controls. And they looked how individuals who were diagnosed with the first clinical event, so clinically isolated syndrome, used the healthcare system in the preceding 5 years and showed that the use of healthcare was highly significantly increased in individuals with a prodrome who would eventually develop multiple sclerosis. So, and this is true in adults and in children. So people have these symptoms and signs of that are very non-specific, but contrary to most people who experience them, it's got to be significant enough for them to go to emergency room department and actually look for help. So this is interesting, but of course, currently it doesn't really translate into anything actionable. Again, you have people with these very non-specific signs and symptoms, and usually we don't diagnose them with anything, and we certainly don't treat them with anything because there's no biomarker, there's no diagnostic marker that would allow us to do this. And I think that is about to change. So the first biomarker that has really been evaluated in the last few years is shown on this slide that I showed earlier, neurofilament. So neurofilament is a scaffolding protein that is expressed in axons and neural dendrites in the CNS, but also in the peripheral nervous system. And so it's really a plausible biomarker for a disease like MS where axons and neurons are being damaged. In humans, there are 5 proteins in the neurofilament family, and 3 are shown here. These 3 have been studied as biomarkers in multiple sclerosis. There are neurofilament from the top, heavy chain, medium chain, and light chain. And the idea is that you have an MS attack or an injury to an axon. The axon dies, neurofilament diffuses out into the extracellular space, and then into the cerebrospinal fluid and into the blood, where you can detect it. And I circled neurofilament light chain because it's the smallest of all the neurofilaments, and with a protein that's small, it's easier to diffuse into the tissue. So it's really been a better biomarker than the larger proteins. And I should say that since 2000, there's an ELISA test of the CSF, the cerebrospinal fluid, that's actually approved in Europe and North America, but it really hasn't been used much. And I think that's a cultural phenomenon, at least in the United States. Patients don't want to undergo a lumbar puncture, and they certainly don't want to undergo serial lumbar puncture. So it really hasn't been used. So in 2018, a new assay was introduced that's essentially also an ELISA. It's called a single molecule array, or SIMOA, and people call it a digital ELISA, where you have the same components. You have a paramagnetic bead, in this case with a capture antibody that captures a protein like neurofilament light chain, and then you have a detection antibody with a fluorochrome, and you can read it. The difference is that you only have one paramagnetic bead per well, and so on the lower right bottom of the panel, you can see that if you look at a 96 well plate, for instance, you either have the protein or you don't have it. That's why, so it's binary. That's why they call it a digital ELISA. And it's 1,000-fold more sensitive than a traditional sandwich ELISA, and it's now approved to test for neurofilament light chain in blood. So that's a real step forward. The other advantage of a SIMOA over a sandwich ELISA is that it's easy to use. So I don't know if anybody has ever used a sandwich ELISA. It's not a complicated assay, but it's a lot of steps, a lot of washes. It's highly user dependent. The SIMOA assay, it's one drop of blood, so like a little finger prick or an earlobe prick. You put it into a well, you stick it into a desktop machine, you push the on button, and pretty much that's it. So any clinical personnel in an emergency room or in a clinic setting, a nurse, a doctor, can really use this. So that's really a step forward. Now, of course, I mentioned to you neurofilament light chain is a scaffolding protein expressed in all neurons. So it cannot be a MS-specific marker of disease, right? Because any disease that causes injury to neurons in the brain or in the periphery, including diabetic polyneuropathy, is going to change your serum neurofilament level. So it's a good first step, but it's certainly not a disease-specific step. And for this, there's another new method that you may or may not have heard about. I'm very excited about this, and I know it's going to become available everywhere in the world. It's called an Olink assay. Now, some of you may know flow cytometry. Flow cytometry is essentially an assay where you, again, have a protein and a detection antibody that's labeled with a fluorochrome. And you know, if the antibody detects the protein, you can basically measure the fluorescence with an apparatus. The problem is that fluorochromes have overlapping light spectra, so you're always limited to 15 proteins that you can detect at one time. The Olink assay is a real step forward, where the detection antibody is actually labeled with a single-strand DNA barcode. And you use two detection antibodies against two different parts of the protein. And if both of those antibodies attach to the protein, the two single-stranded DNAs anneal, and you can detect it with a PCR method. And because there are really indefinite numbers of DNA barcodes, you can detect indefinite amount of proteins. So with one drop of blood currently, you can detect 7,000 proteins, which is really quite amazing. And Olink is also the name of the company that developed this assays, but there are now a lot of companies that use assays very similar to this that have become commercially available. Now, what do you do with 7,000 proteins? It's hard, of course, to make sense of all of this. And there are companies that do that. There's one company called Octave Bioscience in Silicon Valley, and they did something very clever. They used the MRI disease activity in patients with MS as their gold standard. So specifically, they looked at MRI scans that had contrast enhancement. And then they took blood in various intervals before the onset of the contrast enhancing lesions, at the onset of the contrast enhancement, and afterwards. And they took blood and used this Olink assay. And then they used AI, artificial intelligence, to really narrow, these thousands of proteins down to a reasonable amount of proteins that correlated with the onset of these lesions that enhance with contrast. So basically, they came up with a panel of 18 proteins that really predict the onset of contrast-enhancing lesions. And that is now basically being used in many healthcare systems in the United States, and I think it will replace MRI scan, quite frankly. It's going to be a lot cheaper. It's going to be much more easy to do. It's going to be standardized across centers. So again, I think that's a real step forward. We are doing an investigator-initiated phase 4 trial. It's really a trial that was designed to do immunological studies, where we enrolled 40 patients who were on a drug called natalizumab. They were all transitioned to another drug called cladribine. But we used the trial to test, we were actually the first one to test this Olink assay by Octave Bioscience prospectively. And this is a poster, you won't be able to see anything, but basically over 24 months, the Olink assay perfectly correlated with clinical disease activity and MRI activity in this prospective trial. What's more is that some of the patients in this trial had been diagnosed with relapses and the Octave biosignature didn't change. And we were really curious about that. And then we went back to these patients and it turned out they didn't really have an MS relapse. They had an infection typically of the brain or the spinal cord with a herpes virus. So it's not only very sensitive and correlates with disease activity, but it's really also MS specific. So I think that's what's going to help us early on when you have patients with a prodrome or radiologically isolated syndrome to identify individuals who are at risk for eventually actually developing multiple sclerosis. So just to come back to the MS prodrome, currently you will be working in your clinical practice or an emergency room department. Somebody's going to come in or bring in their child and say, something is wrong. I'm itching, I have fatigue, I just don't feel like myself. And currently, you're going to do a history, a physical exam, you're going to maybe do some x-rays or blood tests, and you're not going to find anything, right? We all have been in the situation. You're going to say, I hope you're going to feel better. I don't know what's wrong with you, and come back in 2 weeks if things don't get better. In the future, I think you're going to be using things like Simoa assay, Olink assay, and I think this is not just multiple sclerosis. I think it's for many, many diseases that we're using right now, and I think you're going to figure things out. And again, if you look back at this map, I think Olink is also going to change how in countries that are currently under-resourced, to make these diagnoses, will be able to do so and really provide care for their people. Now, what causes multiple sclerosis? I'm briefly going to talk about that because that's a huge enigma. I always say it's an autoimmune disease, and that's what most people think, but we really don't understand the pathogenesis completely. Genetically, there is one gene that's really established to be associated with the risk of developing MS, and that's an MHC class 2 gene. In humans, it's HLA, specifically HLA-DRB 1501. And it's biologically important because it explains inflammation. So you have here an antigen-presenting cell on the left, and that's typically a dendritic cell or macrophage or B cell, and they express a molecule called MHC, in this case MHC class 2, and that's why they're called antigen-presenting cells. And the MHC molecule has a groove that can hold a linearized peptide, which is an antigen, and then if a T cell, in this case a CD4 T cell, with a T cell receptor specific for this antigen, sees this antigen in the context of the MHC molecule, it becomes activated and it differentiates to become pro-inflammatory. So the T cell can only see an antigen if it's presented by an antigen-presenting cell in the context of an MHC molecule. That's very important. If the peptide was flying all around the T cell, but there wasn't an antigen-presenting cell, it would be blind to it. This is called the trimolecular complex, the APC, the T cell, and the antigen. There are 210 or so other risk alleles, genetic risk alleles associated with MS, but they all have a very low odds ratio. And frankly, the genetics only explain 30% of the risk of developing multiple sclerosis, roughly. So it's a complex genetic disease, and there need to be other explanations why patients develop MS. And the current thinking is that there are people that are genetically susceptible, but there need to be environmental factors that trigger the disease. And the most well-established environmental risk factors currently are vitamin D deficiency, tobacco smoking, the use of makeup, and infection with a pathogen. And most of us think it's probably a herpes virus. And some of you may have heard about this publication by Alberto Osheria's group at the Harvard School of Public Health that was published in Science 3 years ago. And they really showed a very, very strong association between the onset of multiple sclerosis and an acute Epstein-Barr virus, EBV infection. So they looked at the healthcare records and blood samples of 20,000 US military personnel. If you're in the US military, everybody has to give blood every 6 months. So it's really a treasure trove for people who like to do research. And they looked over the course of 20 years, and there were 901 military personnel who had a diagnosis of MS. During that time, there were 38 who actually developed multiple sclerosis. And in all but one of those 38 individuals, in the 6 months before they developed MS, they were EBV negative. And when they developed MS, they all became EBV positive. And that's a pretty strong association, not quite causality, but definitely a strong association. And of course, EBV is very prevalent. I don't know about South Africa, in most countries it's between 80 and 90%. So it's still hard to figure out, again, why some people develop MS and why others don't. One mechanism that's being entertained is molecular mimicry, and that's something I think you all also have heard about. It basically means that you have a pathogen that's different enough from autoantigens to be recognized by your immune system. It's a danger signal, but it's similar enough to an autoantigen to induce an immune response against the pathogen, but also against the autoantigen. And typically it's a two-step event where you have, let's say, an EBV infection. So there's an activation of the immune system and formation of immunological memory against the pathogen, but also against the autoantigen. So if you have a second infection, there's an amplified immune response, again, against the pathogen, but also against the autoantigen. And that could cause autoimmunity. And autoimmunity really exists in multiple sclerosis. That was actually discovered 25 years ago unintentionally in the context of a clinical trial. This is work by the NIH that was published in Nature Medicine in 2000 by a colleague, Bibi Bilyukova. And what they did was they tried to develop an antigen-specific therapy for multiple sclerosis. And that is called an altered peptide ligand. And it uses this concept. You basically introduce a peptide that looks like the one that causes multiple sclerosis, but you modify it to change the affinity and the avidity of the T cell receptor to the peptide. And what happens is that this T cell no longer becomes activated, but actually becomes anergic. So it becomes dormant or even dies. It didn't work, however. The trial had to be stopped after just a few weeks because 7 patients developed horrible MS exacerbations. You can see 3 of these individuals here, where the top panel is always the baseline MRI scan, at the bottom panel is the 7-week MRI scan, and it shows you that this is molecular mimicry. You make something that looks just like an autoantigen, but it's different enough to be recognized as a danger signal, and then everything goes haywire. And that is something that's being entertained for EBV. there's a group at Stanford, Tobias Lance, who published a Nature paper just a week after the Science paper with EBV came out that showed there is molecular mimicry between an EBV protein and a human adhesion molecule in the brain that I never had heard about, glial cam, and that is something that's now being basically investigated in other centers around the world. There's also, this has already translated to therapies. Moderna, which is a company that makes mRNA vaccines, is doing one trial with an EBV vaccine to prevent MS that enrolls more than 100,000 people currently. And they also do a treatment trial with a vaccine to see if they can treat multiple sclerosis. How much time do I have? Do I have another 10 minutes? So again, another question is, why do people transition from early MS, which we can treat, we have 24 therapies for it, to secondary progressive MS, which we can currently not treat? I told you that there's inflammation in all stages of multiple sclerosis. This inflammation, at least early on, we know is driven by leukocytes coming from the bone marrow. So one therapy that was introduced and approved 20 years ago is a monoclonal antibody called natalizumab. The brand name is Tysabri. It blocks an adhesion molecule called alpha-4 integrin and its interaction with its ligand BCAM1 on the blood-brain barrier. So in essence, it prevents leukocytes that come from the bone marrow to get into the brain and the spinal cord. And that's shown here. So T cells become activated somewhere in the body against a CNS autoantigen, but they cannot get into the tissue. And we actually showed that 20 years ago, where we showed in CSF of patients treated with natalizumab, the number of leukocyte subsets is roughly 50-fold lower than in people treated with other therapies. And the manufacturer of natalizumab first tested natalizumab in the early phase of multiple sclerosis, in relapsing remitting inflammatory MS, in 2 phase 3 clinical trials, and I'm not going to go into details. It was phenomenally effective. And everybody who treats MS and uses natalizumab will know that in many patients it's like a cure. They will no longer have any disease activity. The drug has significant side effects, but it's so good that a lot of patients just insist on staying on it. So emboldened by these results, the manufacturer said, okay, it worked so well in early MS, we're going to use this in secondary progressive late-stage MS. And they did a huge phase 3 trial that was placebo-controlled and was completely negative. So once patients transition to secondary progressive MS, a drug that is almost like a cure in early MS becomes completely useless. That shows you the biology is completely changing. So whereas in early MS, as I stated, leukocytes come from the bone marrow and enter the brain and the spinal cord and cause inflammation, in secondary progressive MS, that's no longer necessary. So the input of peripheral cells is completely unnecessary. But inflammation is still there in secondary progressive late-stage disease. So all the components that sustain this inflammation in the brain spinal cord are already there in the CNS. And Danny Reich, a colleague at the NIH, and others have shown in the last 10 years that something interesting happens in secondary progressive MS. And those are called smoldering lesions. And that's shown on the panel on the left. It's like a granuloma, if you look at it. You have an acellular kind of necrotic core surrounded by a rim of macrophages and microglia cells. And many of these cells are phagocytes, they're all activated. And these lesions expand and slowly destroy tissue. And there are really good data now showing that these rim lesions are really what drives neurological disability in multiple sclerosis. What's great is we can image these in real time as they occur and then evolve in patients with secondary progressive MS. And just real briefly, we wanted to know why Tysabri or antagonism of alpha-4 integrin does no longer work if these myeloid cells are so important in these rim lesions in secondary progressive MS. So we basically made an alpha-4 integrin knockout mouse and induced EAE, which is the animal model of multiple sclerosis, and showed yeah, these mice, even though you knock out the most important adhesion molecule that allow bone marrow cells to get into the CNS, they still develop disease, same incidence, same severity. And what we showed was that if you knock out alpha-4 integrin or you antagonize it with a monoclonal antibody, a subset of macrophages will upregulate other adhesion molecules that will allow them to get into the brain. So that's probably why Tysabri doesn't work, or nalolizumab. Also, if you think about, let's say you're CEO of a big pharmaceutical company, you want to know these rim lesions, are they really microglia cells, CNS intrinsic microglia cells, or are those macrophages that come from the bone marrow? Because it changes how you develop a pharmacological intervention. And that's something we also studied. It's easy to do this in mice. We made a bone marrow chimera where donor and recipient are genetically identical, except for one congenic marker on CD45. CD45 is a marker for all leukocytes in the body, but the donor has CD45-1, the recipient CD45-2. So in the brain, you know everything that CD45-1 is from the bone marrow, everything CD45-2 is the microglia. And then you can do transcriptomic analysis and so forth. And we showed that these cells are very, very similar, macrophages from the bone marrow microglia, but there are transcriptomic signatures that help to differentiate them. And that's, of course, something that you could use also to devise specific interventions for one or the other. In the last 2 minutes, I just want to say these smoldering lesions, of course, will also hopefully help to find new diagnostic markers for progressive MS, because, as I said, there are MRI sequences that help us to visualize them. And as was done with early relapsing remitting MS, you can now correlate the occurrence of these lesions with serum biomarkers and find a signature that will predict the transition from early MS to secondary progressive MS. And more importantly, knowing that these lesions are there and what they're constituted of, you will be able to devise therapies for patients with late stage secondary progressive MS. And that's already happening. There is a family of drugs, they're called Bruton tyrosine kinase inhibitors. They're already approved for B-cell lymphomas and other kind of hematopoietic malignancies. They deplete B cells and they modulate myeloid cells to become less inflammatory. I'm going to skip this, but this was just published in the New England Journal of Medicine. One of these drugs is tolebrutinib. It was tested in late-stage MS in patients who no longer have relapses and MRI lesions, and it prevented the worsening of neurological disability. And actually, it's thought that in this week or next week, the FDA will approve it as a first drug for late-stage secondary progressive MS. So with that, I just want to say a lot of things are happening in multiple sclerosis, a lot of biomarker research. I think that will completely change how we diagnose multiple sclerosis, how we monitor multiple sclerosis, how we monitor therapeutic interventions in multiple sclerosis. So I think it's a very exciting time to be part of that. As I mentioned, a lot of these assays will be commercialized. I think they're going to be very inexpensive. So I think that's also going to change how we diagnose and manage multiple sclerosis in countries that currently don't have full access to everything that's available. I showed you a little bit of data. A lot of people were actually involved with this, both at UT Southwestern, but also collaborators around the world. These are some of the lab members currently. We went out for dinner 2 weeks ago. I want to thank everybody in the lab. And of course, I want to thank all my friends. This is Lisa and I 31 years ago. And Lisa has always inspired me to do my best. So I'm very fortunate to have her as a friend. And I want to thank you for your attention. Thank you very much.
Awful picture. So that was absolutely brilliant. I totally love that. I think some people often ask me, why do I work at the MRC? Because I'm a pediatric cardiologist and they don't do any pediatric cardiology. And it's for exactly this reason, is that you are introduced and see these stories of why science is so amazing. The story of how you make the diagnosis, how you get to the translational part, how you can actually open up the whole field. Whereas 25 years ago, 30 years ago when we met, there was no therapies.
Mm-hmm.
I mean, this is just a remarkable story and so beautifully done. So thank you so much for that. I thoroughly enjoyed myself. And I see we've got quite a few people on online. I'm asking Ismail to look because I didn't bring my computer. I just want to check whether there are any questions from within the room, and then we can go online. Any questions? Robbie, lovely to see you.
When you score with the RIS and you identify using the IRS, and you said only 50% went on 10 years later to develop the disease. But then you also mentioned with the Olink assay that you've done some clinical trials and that you have 18 markers that you use and it matched and it was able to identify. When you look at individuals maybe that in which with the RIS, those that do develop and don't, were you able to see those same markers in there? And then my second follow-up onto that would just be, you're speaking about gene-environmental interaction. Have you ever given thought of the role that methylation plays in this whole process?
No, that's a good question. I'm just going to repeat it. So the question was whether in the trials that were conducted in individuals with RIS, any of the assays that I mentioned were used to distinguish patients at risk of developing MS versus not. And the answer is no. You know, the timing was really bad. These trials were conceived roughly 7 years ago and enrolled very quickly. And for instance, the Olink assay wasn't available, or the people who conducted the trials weren't available of the Olink assay. So they did trials where they looked at inflammatory cytokines like interferon gamma, TNF alpha, those kind of things, but there was no difference. And I think that's kind of thinking of 30 years ago, you know, that 1 or 2 cytokines drive a disease like multiple sclerosis, and I think it's really oversimplified. It's much more complex than that. So I think it will really take an assay like Olink or mass spectroscopy to find the difference between individuals at risk or not at risk. You know, and it may even be more complicated than that. Like, when do you look? You know, because these inflammatory markers may only be expressed for a very, very short time, during which everything is set in motion and then no longer be detectable in blood. So I think there's still a long road ahead, you know, to provide us that kind of clarity. Yeah. So at this point, no, we don't.
And methylation? Have you ever thought to look at some of, if you identified susceptible genes and the role that methylation plays in it, especially when you mention about gene-environmental interaction that could possibly play a role?
Yeah. So I didn't understand the entire question, but yes, people are looking at gene-environment interactions. So there are centers that have a lot of genetic data, like UCSF and MIT, Harvard. And they look, for instance, how does genetic profile and vitamin D deficiency play together to enhance the risk? And there are actually good published data on that. That if you are genetically susceptible and you have vitamin D deficiency, it's much worse than not having vitamin D deficiency. The genetic data is complex, as I said, and we're not testing genetic profiles. So HLA-DRB1-1501, for instance, is so frequently expressed in North America and Western Europe. A third of the population has at least one allele. So it really wouldn't make any sense to look for that, you know. So I think that's also something that we don't fully understand, you know. Vitamin D signaling is affected actually by your HLA genotype. That's also published, but that's only one environmental risk factor. We do know that there are really good models, mathematical models of environmental risk factors. Doug Gooden is somebody who published extensively on that, and we know that one environmental risk factor, for instance, is not enough to cause autoimmunity. You have to have at least 3, very likely, you know. Yeah.
Any other questions from yes, Kim, go ahead.
Thank you very much for the lifetime word. I'm just wondering if do you have any data for HIV populations? Is also in the longitudinal study that looking into the military populations. And so I just also wondering that if stress play a role in terms of triggers the development of MS over the life course. Mm-hmm. And eventually someone develop disease and someone not. And because I also think of due to inflammation effects. Mm-hmm. So the lifestyle in general, but also maybe mental health, especially stress. Have you got any data on that? Mm-hmm. Yeah.
Yeah, so the second question, there are data showing that psychological stress is a risk factor for relapses, you know, and so is surgery, for instance. People have an increased risk of having MS relapses right after surgery, major surgery, I should say, you know. And that kind of makes sense because those are all events that can activate your immune system. And if you have an aberrant immune response against an autoantigen, it would make sense that these events really increase your risk of having a relapse. Any febrile event increases your risk of having an MS relapse. HIV, it's interesting. So before antiretroviral therapies were introduced in 1997, there was a lot of work on HIV and multiple sclerosis, most of it done by somebody called Joe Berger at the time at the University of Miami. And it was interesting because they showed that the risk of MS is the same in individuals infected with the HIV virus and actually in individuals who had full-blown AIDS compared to age-matched controls, which was unexpected at the time because MS was really thought to be driven by CD4 T cells, right? So if you have a disease that completely depletes CD4 T cells, how can people still develop MS? But it happened, you know, and I think what we learned was that you need actually very, very few T cells to cause autoimmunity. And of course, HIV doesn't just deplete T cells, it does a lot of other things like it infects microglia cells. But overall, it doesn't affect the prevalence or the incidence of multiple sclerosis. And I actually don't know the literature post 1997, you know, when antiretroviral drugs were introduced, but I haven't heard anything. So I don't think they have changed that.
I have two questions from online, and so this first question comes from Antonio Lento. He's external to the SMRC, so maybe from one of the universities. His comment is, I know this is not the focus of the presentation, so you've been put on the spot here, but how do you feel, what do you see the role of neuropsychologists and cognitive testing in early MS, especially around the subtle cognitive changes potentially as a biomarker, and then guiding earlier intervention with disease-modifying therapies?
Yeah. That's a really, really excellent question. And actually, it really is part of multiple sclerosis and managing patients with multiple sclerosis. So one thing I didn't talk about was how do we measure neurological disability? And we're not good at it, you know? So we use a scale called the EDSS scale, which really heavily focuses on the ability of a person to ambulate. So, you know, we measure how fast they can go, how far they can go, do they need unilateral or bilateral assistance, those kind of things. But MS disability is much, much more than that, including cognitive disability. And in fact, we know that 90% of patients will eventually develop substantial cognitive disability. In the healthcare system where I work, it's called the VA, you know, the Department of Veterans Affairs, where we see veterans of the US military. We actually can test cognition every 6 months, and that's very helpful to see how patients are doing. That's not covered by Medicare and private insurance. On the outside, that's not being done. But I think there's now a recognition that we should focus on non-physical disability to really monitor patients, but also to monitor therapies. You know, are they actually benefiting individuals. So yes, I think it will hopefully become part of our clinical assessment.
Another question here about biomarkers. So do you think that biomarkers that respond to environmental variables could potentially be used as markers of therapeutics of the human immune response?
Could you repeat that, please?
So biomarkers that respond to environmental variables, could they be used as markers of therapeutics of the response?
Maybe yes. So, I mean, if you have environmental variables and they're disease specific, I think they will be used, you know, to measure therapeutic responses. And that's something we still don't know. So for instance, is it completely random if you're exposed to a biomarker, whether you develop multiple sclerosis or rheumatoid arthritis or another autoimmune disease? Do they all have the same environmental risk factors? We don't know that. So I think eventually there will be a composite of biomarkers specific for each autoimmune disease. And I think they will be used then to monitor disease activity.
Fantastic. I'm going to take the opportunity, sorry to put Shelley on the spot, introduce you to Michelle Mulder, who is part of our Executive Management Committee, works in our GRPD area. And I just thought the concept of things like we're looking at point of care diagnostics, and I thought the O-link idea sounded like fantastic, but also the issue of getting them into low and middle income countries. Often the reason why we don't fit onto a map is because there's no diagnostics, rather than that it doesn't happen in our country. And I just wondered what you thought about that, Shelley.
Yeah, and it sounds like this O-link has got quite a lot of application beyond, obviously, MS. If you're looking for did you say 700 or 7,000?
7,000.
7,000 proteins. So what it just means is somebody has to find the algorithm for the rest. And, you know, that's the kind of platform technology that I think has got a lot of application in LMICs, but it depends also on the fancy equipment that comes with it. I saw you had a picture of a piece of equipment. I don't know if that's just a standard piece of equipment or if that's very specific to this kind of assay. When you say you push a button and this spits out the answer, which often means a very specialized piece of equipment, and that's often the barrier, because that's where the cost comes in, not just to purchase and roll out, but also to maintain and support that equipment. So yeah, but I think the other thing to point out though is this. What we should be doing a lot more of is the early screening, particularly if you can do did you call it the ProDiam, where prevention is better than cure, and you can stop a disease before you get to the very expensive treatment options. You know, biologicals in this country are out of reach of most of the population. So we have to stop these diseases before they even start, or catch them very early on. So, but the problem with that is you're spending money for something that you don't know if it's going to have the impact, because you're having to screen the whole population just about to catch just those few who might progress to disease.
I should say a lot of data can be analyzed already. So, for instance, every research project that's funded by the US federal government or the Canadian government or the German government or the UK MRC is publicly available, you know, so a lot of data is out there to be analyzed. And, you know, young people love coding and they love AI. So we have students who are great at doing that. And quite frankly, there's an explosion of knowledge, you know. So even ChatGPT-5 is terrific for analyzing biological data. And so I think we will no longer depend on a company like Octave Bioscience to do this, you know. You're going to have some smart people in your lab that will be able to do this, you know. So, and again, a lot of data are publicly available. People just don't know it, you know.
I think we're going to wrap up. I think there was one more question. Yes,
thanks.
please.
I think you answered my question. I wanted to ask about the low prevalence of MS, whether it's due to poor diagnosis or due to the genotypes of our population. Since you said the HLAs that increase the risk of MS are more in the European populations.
The prevalence in South Africa.
Oh,
alone.
yeah. So the great thing about these whole genome scans, I think the main observation is that genetically we're all the same, So it's the same, like for instance, MS in Asia, in Japan, and China. The prevalence is different. It's not genetics, because we're literally genetically identical, So it's got to be either environmental factors that are different, or really just the ability to diagnose a complex disease like that. And of course, the prevalence of MS changes, and the best data for that actually comes from Iran. They also have a nationalized healthcare system, and they showed that after the revolution in 1979, the incidence in women went sky high. And they think it's because they had to cover themselves, so they developed vitamin D deficiency. They started smoking more. I don't know why they started smoking more after the revolution. They used more makeup. I don't know. I can't tell you why they started doing all these things, but that's actually how a lot of these biomarkers were discovered, So it's not a static thing. I think in every population, the risk of these autoimmune diseases changes constantly with these trivial, environmental factors. Yeah.
Fascinating. So just as you said that, somebody asked about the difference in gender. So you've answered that, and I think that's really interesting. I'm going to wrap up now, simply by thanking you so much. Olaf is here on a holiday. He's certainly not here to be giving lectures. So we really appreciate your time and for telling us this wonderful story, but hopefully also for inspiring all of us to think a little bit out of our usual boxes. To think about translational science, impact on patients, and also using things in a new way. I think maybe your comment about ChatGPT-5, how we can actually use the tools available to us in this country to be able to do experiments that we think we may not be able to do because of the cost relations. And I think that is something that can only be an inspiration. So thank you so much. We really appreciate it.
Thank you
very much.
Thank you very much.
Adam loves to take a photo.